Extracting dino blood from mosquito amber was hard enough, so how on earth did they get hold of Mosasaurus DNA? This was the question that puzzled many when InGen, the pioneering biotechnology company, unveiled their latest attraction, the Mosasaurus. Unlike the terrestrial dinosaurs resurrected through blood extracted from amber-trapped mosquitoes, the aquatic Mosasaurus posed an entirely different set of challenges. Join us as we delve into the fictional methods employed by InGen, as well as the theoretical, real-world science behind recreating such a magnificent creature. Oh, by the way, you had one job, InGen, or should I say Masrani Corp. Stop the dinosaurs from escaping. Again. I mean, raptors getting loose is somewhat forgivable, but how did a 60-ton sea creature manage to escape the park? I see you built a very, very thin partition between the aquarium and the ocean. I mean, why? But first, let's discuss the InGen method. In the year 2000, InGen researchers faced a daunting challenge. They aimed to extract DNA from a Mosasaurus, a marine reptile that roamed the late Cretaceous seas. The traditional method of obtaining dinosaur DNA through blood meals taken by mosquitoes and preserved in amber was not applicable to marine animals like the Mosasaurus. Instead, they needed a different approach. While testing a prototype iron analyzer on a recently uncovered Mosasaurus fossil specimen, InGen researchers made a groundbreaking discovery. They detected traceable DNA fragments within the fossil. This method bypassed the need for amber-trapped mosquitoes and allowed direct extraction from the fossilized remains. Dr. Henry Wu, the brilliant geneticist at InGen, saw this as the key to recreating aquatic organisms. With the DNA fragments in hand, InGen scientists faced the challenge of reconstructing the complete genome. The DNA retrieved from fossils was fragmented and incomplete. To fill in the gaps, they used the DNA of modern marine animals, such as sharks and crocodiles, which are genetically similar to the ancient Mosasaurus. This process of combining ancient DNA with that of modern animals allowed them to create a viable genome. Once the genome was complete, the next step was cloning. The reconstructed DNA was inserted into an egg cell from a closely related modern species. The egg was then fertilized and developed in a controlled environment until it was ready to hatch. This marked the birth of the first Mosasaurus in millions of years. The newly created Mosasaurus, dubbed Mosasaurus maximus, became the star attraction of Jurassic World. Housed in a 3-million-gallon pool near Main Street, the Mosasaurus thrilled visitors with its feeding shows, where it leapt from the water to catch a shark. This spectacle showcased the power and majesty of the Mosasaurus, captivating audiences worldwide. The Mosasaurus in Jurassic World is estimated to be around 60 feet to 120 feet in length, depending on various scenes and promotional materials, with a weight in the 40 to 60-ton region. While the methods depicted in the Jurassic World franchise are fictional, they are based on real scientific principles. In this section, we explore how a Mosasaurus could theoretically be created using current and emerging scientific techniques. In the real world, extracting viable DNA from ancient fossils remains a significant challenge. DNA degrades over time, and the oldest DNA sequences recovered to date are from specimens that are less than 1 million years old. However, advancements in ancient DNA technology continue to push the boundaries. The advent of next-generation sequencing technologies has revolutionized the field of genetics. These technologies enable scientists to sequence even highly degraded DNA by piecing together millions of small fragments. By applying these techniques to Mosasaurus fossils, scientists could potentially reconstruct parts of the genome, although it would still be incomplete. To create a complete genome, scientists would need to fill in the gaps with DNA from related species. In the case of the Mosasaurus, the closest living relatives are monitor lizards and snakes. By comparing the Mosasaurus DNA fragments with the genomes of these modern reptiles, scientists could identify homologous sequences and infer the missing parts of the Mosasaurus genome. Synthetic biology offers another avenue for recreating extinct species. Once the genome is reconstructed, scientists could use genome editing tools like CRISPR-Cas9 to insert the reconstructed DNA into the genome of a closely related species. This would allow the host organism to develop with the genetic traits of the Mosasaurus. Assuming the genome reconstruction is successful, the next step would be to develop the embryo. This involves inserting the edited genome into an egg cell from a compatible modern species, such as a large marine reptile. The egg would be fertilized and allowed to develop in a controlled environment. This process, known as somatic cell nuclear transfer, has been used successfully to clone animals such as sheep and dogs. While the theoretical steps to recreate a Mosasaurus are grounded in real science, several ethical and practical considerations must be addressed. Cloning an extinct species raises numerous ethical questions about animal welfare, ecological impact, and the potential consequences of introducing such a creature into the modern world. Moreover, the technological and biological challenges are immense. The degradation of ancient DNA, the complexity of genome reconstruction, and the difficulties of cloning large marine reptiles are significant hurdles that may take decades to overcome. The concept of de-extinction, or bringing extinct species back to life, is a subject of intense scientific interest and debate. While recreating a Mosasaurus may remain within the realm of science fiction for now, advances in genetics continue to make strides toward the potential revival of other extinct species. Current de-extinction efforts focus on species that went extinct relatively recently, and for which good quality DNA samples are available. For example, scientists are working on projects to bring back the woolly mammoth, passenger pigeon, and Tasmanian tiger. These projects provide valuable insights into the challenges and possibilities of de-extinction. One of the primary motivations for de-extinction is the potential to restore lost biodiversity and strengthen ecosystems. By reintroducing extinct species, scientists hope to fill ecological niches that have been left vacant, thereby promoting environmental stability and resilience. However, the success of such efforts depends on a thorough understanding of the ecological roles of these species and careful planning to ensure that their reintroduction does not disrupt existing ecosystems. As technology continues to advance, the possibility of recreating extinct species becomes more feasible. Improvements in DNA sequencing, genome editing, and synthetic biology hold the promise of making de-extinction a reality. However, each step forward also brings new ethical considerations and challenges that must be carefully navigated. The idea of bringing extinct species back to life captures the public imagination, as evidenced by the popularity of the Jurassic Park franchise. However, public perception is divided. Some see de-extinction as an exciting scientific frontier with the potential to right past wrongs, while others view it as an unnecessary and risky endeavor that could have unforeseen consequences. Ultimately, the pursuit of de-extinction requires a balance between scientific curiosity and ethical responsibility. It is essential to weigh the potential benefits against the risks and to engage in open, transparent discussions with the public and the scientific community. The goal should be to advance our understanding of genetics and ecology while ensuring that any actions taken are in the best interest of both the species involved and the environment. The fictional creation of the Mosasaurus in Jurassic World is a testament to the power of imagination and the allure of bringing the past to life. While the methods depicted in the films are grounded in real scientific principles, the actual realization of such a feat remains a distant goal. Nevertheless, the ongoing advancements in genetics and synthetic biology continue to push the boundaries of what is possible. I hope you enjoyed this Mosasaurus documentary. Please consider subscribing for more dinosaur documentaries. And if you wish to purchase the dinosaur mugs, the links are in the description. Until next time.